Okay, well, good evening everybody, and welcome to the 6th annual Environmental Microbiology lecture. And this lecture, as you know, is held to celebrate the success of the journal Environmental Microbiology under the editorial leadership of Ken Timmis and his outstanding editorial team. Tonight's speaker, Victor de Lorenzo, is a chemist and a microbiologist. He's Professor of Research at the National Centre of Biotechnology in Madrid, where he's been since 1996, but he's previously worked at the Institut Pasteur in Paris, the University of California in Berkeley, the University of Geneva, and the Federal Centre of Biotechnology in Braunschweig. His research exploits advanced molecular biology and genetic engineering of soil microorganisms such as Pseudomonas putida, for the sake of biomonitoring, bioremediation, and wherever possible, valorization of chemical pollution in the environment. He's a member of the European Molecular Biology Organization, the American Academy of Microbiology, and the European Academy of Microbiology, and the jewel in the crown is also a member of SFAM. And he's going to talk to us this evening on programming soil bacteria to do amazing things. So without more ado, I'll hand you over to Victor.
Good afternoon, ladies and gentlemen. Thank you, Martin, for this very friendly and kind introduction. So I have to admit that I never or very infrequently have an audience of this stature. So when I was coming here, I was recalling something that I read in a book that was written by a Jewish 12th century philosopher called Maimonides. The book was called A Guide for the Perplexed. So he said, there are 3 levels of communication between a speaker and an audience. The best, the most perfect, is the one in which the speaker knows what he's talking about and the audience understands. There's another level in which the speaker knows what he's talking about, but the audience does not understand. And there's a third level in which the speaker does not know what he's talking about and the audience cannot understand. So I hope that by the end of this talk, we will qualify in the first category rather than in the third one. All right, so programming soil bacteria to do amazing things. This is the center where I work, a National Center for Biotechnology in Madrid. And before I forget, let me just show you the faces of my collaborators as the laboratory stands now. Some of them, by the way, are sitting in the audience. I will mention some names as I proceed with my presentation. Now, soil bacteria. If you are interested in cemeteries and you visit the grave of Selman Wasman, there's a gravestone with inscription, biblical inscription, the earth will open and will bring forth salvation. And well, this is connected to discovery of streptomycin, and it is very obvious that from soil we have isolated bacteria that do amazing things. Formerly, one of the most amazing products were antibiotics. But what this gentleman could not dream or could not imagine is that many years later, we had discovered such an amazing wealth of interesting products in different activities, different microbial performances, and so on. So soil that you pass by and you think that is dirty, smelly, and everything, it contains a tremendous richness. And it has been our mission for many years to try to uncover this wealth and trying to put it to work in a biotechnological context. We're interested in one specific type of soil, that is soil polluted by the waste released to the environment by the chemical industry, like the one that you see in the picture there. So if you take a closer look to what is there, you find 2 very interesting actors. On the one hand, you find bacteria, microbes, that have been able to live there and to thrive there on the basis of the molecules that chemical industry has misplaced or has created with new chemical structures in such a way that there is a kind of fight, a war between the bacteria and the new chemicals, and at the end, many of these bacteria are able to develop new abilities, new metabolic activities to use these compounds as carbon sources. And to this day, I find that completely amazing, that bacteria develop the metabolic activity and the biochemical capabilities to learn to eat as carbon sources these compounds that they may have never seen before, and that for us are toxic pollutants, for them in many cases are gourmet food. These chemicals can be just products of petroleum, for instance. They are natural products but have been misplaced by the action of the chemical industry. In other cases, what we have is genuinely xenobiotic compounds, compounds that hold chemical bonds that have not been there before the chemical industry put them there. But then what happens is by using very, very simple microbiological techniques like enrichment cultures, one can end up isolating bacteria that indeed can use as carbon sources silicon compounds that have very, very unusual chemical structures. So this is the bottom line, the starting point of the talk of today. Polluted sites that contain bacteria, contain chemicals, and bacteria have learned to use these chemicals as nutrients, and therefore that means that they have genes, they have enzymes that are doing very, very interesting things. Now, in the last few years, there is a very interesting debate on how the industry will look like in the future, and well, this is one of the different flowcharts that have been produced in the sense that many of the procedures of the chemical industry will have to be replaced by more sustainable biological alternatives, and the idea is to start using as the source of these activities the bacteria that live in soil, whether they can be cultivated or not cultured, and then in some cases we can grow them in the laboratory with a slow growth or with fast growth, and then in some cases the one that grows fast can be reprogrammed genetically as a chassis. I will return to that later, as a genomic chassis where we can put activities back and forth by means of genetic tricks. And then in other cases we have access to DNA, and then this DNA, we may not cultivate the bacteria that host this DNA, but still we can take it to the laboratory, analyze the sequence, and again extract activities and convert them in a fashion that can be implanted in this chassis to produce catalysts. And this is indeed a kind of flowchart that will be very useful in the future. Now, what is our business? What is our contribution to this endeavor? Well, I would argue that we are interested in domesticating bacteria. So there's a long way between just finding wild biological entities and having completely predictable, completely engineered. There's an intermediate step that is domestication. And as I will try to argue throughout my talk, in fact, I think that we are exactly in the transition between bacteria that are domesticated and bacteria that are engineered, but we are not yet at the stage that we can engineer altogether microbes for our own sake. However, we can do many, many interesting things at the interface between domesticated and engineered bacteria. Let me also tell you that by the same token that there are many, many animal species, but only about 50 of them have been domesticated. There are a lot of bacteria, and it's possible that only a few of them can be domesticated. We have all faced problems in the laboratories about bacteria that are difficult to manipulate genetically, or cannot be grown, or have different difficult characteristics. Well, but there are others that can be handled with a tremendous ease. So probably there will be at the end a collection of about 50 platform strains that we can manipulate with a great efficiency, and this will be the microbial equivalents of the animals that we have domesticated in the past. So in our own case, as I said, we are interested in domesticating bacteria that live in the environment and grow on strange, unusual chemicals. And this is a short list of some bacteria that have been isolated over the years that do the job very, very efficiently. There are many bacteria that do interesting things, cannot be cultivated. I will not discuss that. I will mostly concentrate on bacteria that can be grown in the laboratory, growing on exotic carbon sources. The list there is a very short list. If we had a complete list, we will have various meters downstairs. And what you have there is a kind of top 10 or 20 or top 12 bacteria that grow fast on the compounds that you have to your right. And note that many of these compounds, toluene, phenol, 2,4-D, and so on, are really, really bad environmental pollutants, and therefore, again, is a big, phenomenon that these microbes can really grow on these compounds as carbon sources. So in our case also, we have been concentrating our work in the last years, well, not in the last years, but for a long time now, in this specific soil bacterium called Pseudomonas butida KT2440. Let me tell you that the strain or the parental strain of this specific isolate was picked in Japan in the 70s on the basis of being an isolate able to grow on metatoluate. And then later it was discovered that this property was endowed by a plasmid, the so-called toll plasmid. I may return to that later, but then this plasmid at some point was cured. So what we have here is a soil bacterium that has just one chromosome, and as you will see in a moment, just by being isolated from soil and from being used to use as carbon sources unusual compounds, it has developed very, very interesting properties. So this strain was sequenced in the early 2000s, and once you have the sequence, then you can really start determining very, very interesting traits. Not only that, but also you can make models, and I'm sure you are aware that in the last few years there are various ways by which one can just go to the genome sequence, and on the basis of the genomes and on the basis of some parameters that can be measured relatively simply, one can construct a very, very detailed metabolic map on how this particular bacterium grows on this on that compound, and also the metabolic models allows us to really get deep into some metabolic characteristics of bacteria of this type. So for us, what is really, really amazing of this bacterium is what we call the metabolic heart. So animals have a mechanical heart that pump blood into the extremities and so on. Bacteria have a chemical heart, a biochemical heart that pump and distribute various metabolites in various parts of the cells and distribute these resources for various functions. And what is really amazing about Pseudomonas butida is that if you look at the metabolism, you look at this metabolic heart, you find that the entire metabolism is geared to do things that other bacteria don't do. What it does, for instance, is that it resists very, very happily solvents, and this is something that is quite extraordinary. So this bacteria can tolerate having in the medium a high concentration of organic compounds. But also, and this is for us what is the most interesting, and I may return to that later, is that the entire metabolism seems to be geared for production of reductive power, NADH and NADPH. And it is this characteristic that the entire metabolism seems to be more interested to produce reductive power than to actually grow, is what endows this specific strain with an extraordinary potential. And I will tell you a bit later why production of NADPH is so important for the type of applications that we are after. At the same time, if you look at the genomic sequence, you find that this bacterium is able to grow in a large variety of carbon sources, not only glucose or succinate, very boring substrates, but on organic compounds with complex structures that again can be entered into the central metabolism through various pathways. And also, and this is very important, is a strain that has the tag, the label of being generally regarded as safe. So that means that you can work on it in the laboratory, in the industry, you can really manipulate this bacterium without being too concerned about risks that might be associated to other types of bacteria that have the potential of being domesticated as well. But then at the same time, we observe in the genome a number of bad traits. It has a number of undesirable antibiotic resistances. It has a complex surface. It has many things on the surface, LPS and fimbriae and flagella and the other structures that, well, don't look good. It has many prophages. It has 4 prophages that are like parasites and can really make this bacterium a bit unpredictable. Then the metabolism lacks a real glycolytic pathway. It has instead this other type of glycolytic route called Ebner-Dodrov, and again I will return to that in a moment. Also, and this is not good news for some applications, is an obligate aerobe, it needs oxygen to grow, something that may not be optimal for some applications, and also it has bad reputation because it's a pseudomonas, and pseudomonas, the general public normally associates that with infections. Okay, but then here comes genetic engineering and the power of DNA synthesis, genetic tools, and all types of recombinant DNA tricks. And then, ladies and gentlemen, we believe, and I argue strongly, that we can really do much, much better than nature by starting with a natural situation, but take that situation into something that is really, really better than we had before. However, to do that, we need tools. And let me share with you this idea that not all tools are equal. So you can have very primitive tools, and primitive tools will allow you to do primitive things. You can have some improved tools, but then for making real engineering, a real modification of bacteria for given purposes, you need standardized tools, and this is a big difference. I'm happy that I'm saying that in this engineering building, because indeed the big discovery, the big contribution of engineers is standards. So standards allow you things that you cannot do if you don't have standards. So what you have there is an example of one of the most impressive civil engineering buildings that are maintained in the world. Is a Roman aqueduct that happens to be in a city of Spain called Segovia. So you may see that in this aqueduct, from the point of view of the engineering, is amazing, and it could be built because the size of the stones were standardized, the size of the arches were standardized, the periodicity of the arches were standardized, and that allowed to go to make a big engineering endeavor that without the standards could not really be made before. Because standards allow the decoupling of the design of an object from the production of the object, of the components of the objects, from the assembly and the deployment. And this is something that we biologists are not used to do. In general, we tend to know or try to control all aspects of a given process, and this is something that engineers say, well, forget it. So we really need to divide the labor and to assign specific tasks to different parts of a process, and this can be achieved only through standards. So just to show you an example of one of the first standards in engineering that made a tremendous difference, and is the standards of the screw terms. Something as simple as determining and fixing the angle, the pitch, the periodicity of screw turns, it really made a tremendous difference, because the design could be made in one place, and then the implementation would take place in a completely different place, and so on. So this is a kind of thinking that we and others are convinced that is going to bring a big revolution to biology, and the first place in microbiology. Because if we standardize all the processes that are involved, or the steps that are involved in the process of modifying deeply a bacterium for a given purpose, then we can really do many more complex, more reliable, and more robust objects, and with more predictable properties. So in the case of bacteria, there are three aspects of the entire manipulation that can be subject of standardization. First of all is the standardization of the functions and the DNA that is involved in the activities that we may want to engineer in a given microbe. Then we need the tools to manipulate them and to deliver these constructs eventually into a destination chassis and in a destination genome. That can be either a natural bacterium, or it can be, again, subject to some refactoring to make it optimal to this end. So let me just tell you on passing that this has been something that has kept us very busy in the last few years, and we are trying to convince our colleagues and the community of scientists and biotechnologists interested in Pseudomonas and in genetic engineering of gram-negative bacteria to really adopt standards, because by doing that we can really amplify the power and the scope of all our endeavors. So one contribution in this direction is the proposition that we have made some time ago to develop what we call a standard European vector architecture. So basically the idea is that the architecture of vectors, plasmids, transposons, you name it, is completely chaotic. Every laboratory uses a different format, a different reporter, a different antibiotic, a different original replication, and at the end it's really difficult to compare results from different places and even to join forces to construct a complex object by collaborating between different groups, because simply different groups have completely different cultures and different, say, ways of assembling DNA parts and so on. So by adopting very, very simple references, like punctuating the structure of a vector with unusual restriction sites, and then use that as a point of reference to exchange functional models, then one can really make a large collection of vectors, all of them standardized, all of them compatible, where one combines origins of replication, antibiotic resistances, and what we call cargos, that are the DNA segments where we engineer new functions. And by doing that, we have been able to make a large collection of plasmids and transposons, as you will see in a second, where we combine, as I said, origins of replication, antibiotics, and different activities in such a way that everything is compatible, everything is coherent, and that allows a tremendous plasticity to the user to make basically anything with Pseudomonas putida and other related bacteria. We have also proposed to adopt a formal nomenclature for plasmids. Everyone gives a capricious plasmid name to whatever construction you make and so on, and this is nice for a while, but then if we are moving into this type of industry where many chemical processes will be replaced by biological alternatives and so on, we really have to take stock from what engineers have made in the past and adopt standards and standards and standards. One can use not only plasmids. If you want to put a gene here or there under the control of your favorite promoter, you can also, you may want also to put things into the chromosome. And for doing that, we have also developed a large series of transposons that will insert your favorite DNA into the genome of Pseudomonas putida or other related bacteria. And basically, we have now all the tools to edit the genome of Pseudomonas putida in such a way that we can change the genes, delete the genes, enter segments, delete segments. It's like a book where you can take sentences and letters and everything and just make a new composition on the basis of what you had before. Okay, so this is available because, by the way, we are strong advocates of open access in the use of genetic tools. So everyone is welcome to adopt these standards and get the vector collection for free, no questions asked. Just only the commitment of following the standards you want to contribute to this vector collection. Okay, so we have the standard, we have a large number of tools. What are we going to do with the tools? Okay, so let me go back to this domestication story. When you want to domesticate an animal, the first thing that one of the first things that you think of is, first of all, to take the parasites out, in this case, phages. Emasculation, so that means that you have an appendix that is not necessary and is really bothering you for some purposes, you can get rid of it. And shaving, so if you have the surface with some hairs or something that you don't like, you may want to take it out. Now, what we have made precisely is to use these tools that I showed you before to do an equivalent operation in Pseudomonas putida. We have taken parasites out, we have taken all the bacteriophages out of the genome, we have deleted the flagellum, for instance, and then we have smoothed out the surface by deleting many of the proteins and many of the functions that give the surface of Pseudomonas putida the type of complexity that we want to get rid of. So this is a procedure that was developed by someone in my laboratory called Esteban Martinez, and he has been able to go up to 5 or 7% of the entire genome of Pseudomonas putida deleted, in which we have really got rid of things that we are not interested to use for the sake of biotechnology, as I mentioned at the very beginning. So what we have now is what we call the surface naked P. putida. Well, in fact, we have a large collection of variants of this P. putida. Remember, at the beginning, the bacteria looked very wild. Now they are more educated, more domesticated. And now the strains that have been deleted of these annoying genes are now sensitive to antibiotics, and therefore that means that that can be manipulated for various purposes. And then the most important for us is that the surface is smooth, and therefore there are many extra manipulations that you can do using the surface as a kind of a scaffold to build things on it. So one of the things, just to show you some of the properties that emerge from these deletions, are really, really beneficial. It has to do with this emasculation that I showed you before. For instance, if you delete the flagella, then what happens is that cells are happier in many respects. So flagella is a structure that consumes a lot of the energy and reproductive power of the cells. We have seen that. And if in reality, if you have the cells growing in a bioreactor, having the cells with flagella doesn't make a lot of sense. You can just shake it up mechanically. And by doing that, you increase some biochemical characteristics of the cells that are very interesting from the point of view of biotechnology. For instance, cells become more tolerant to oxidative stress, and you see in these pictures that when you treat the cells with paraquat or with diamide, two oxidative agents, then cells survive much, much better if they don't have flagella. And the reason for that, or one of the reasons for that, is because we increase the reductive power of cells. So just a note, remember that the way that bacteria have to combat oxidative stress is by producing more NADPH. That is the ultimate metabolic currency that cells have to really get rid of active forms of oxygen or other oxidative agents. So by increasing the amount of NADH and NADPH, then bacteria can tolerate more exposure, more production of oxidation. And this is something that has important consequences for biocatalysis. The other thing that we have observed is that by deleting the flagella, then cells are more resistant or more tolerant to a stationary phase, so they can go for longer. If you let a culture of Pseudomonas putida for a while without carbon sources, up to 10% of the child culture will eventually or soon die, cells will lyse or will lose viability. If you delete the flagella, cells can go for much, much longer. So by doing these operations, genetic operations, they are reminiscent of things that we do with domestic animals. You see that we can really increase the vitality and some important characteristics of Pseudomonas. Now, okay, so we have all the tools, we have the possibility of deleting, inserting, expressing. So what specific endeavors we can undertake? I'm going to show you one case that has kept us busy for some time that goes as follows. I mentioned at the beginning that Pseudomonas putida, one of the downsides is that it cannot grow under anaerobic conditions. Well, it's not that it cannot grow, it's that it dies. When the oxygen is lacking, then cells lose viability. So there are some cases in biotechnology and in bioremediation that we face the following situation. Imagine that we have soil that is polluted with a target chemical that we want to degrade by making a version of Pseudomonas putida that is engineered to degrade that compound. And then the contaminant contaminates the entire soil column and the entire water column. But what happens in reality is that regardless of the distribution of the pollutant, the oxygen will only be on the upper part. So, but we want to have a catalyst that will work on that particular compound, both in the presence of oxygen and in the absence of oxygen. So how can we start with Pseudomonas putida that has a metabolism that is completely aerobic and transform it in something that is anaerobic? Okay, in this case, we just have to look deep into the metabolism and see what can we do. On the one hand, we have to figure out why this strain is only aerobic, and then what choices we have to expand its say, scope of activity under anaerobic conditions. So this was the work of Pablo Nickel that has been working in my laboratory on these topics for a while. And then, first of all, is that when you look at the genes and the metabolic models and everything, then you really see that the genes that are related to anaerobic respiration are absent. So obviously they are absent, so it makes sense that it will not work in the lack of oxygen. However, there are some genes here and there that are related to fermentation, but not a real complete pathway that can be useful. And as a consequence, then P. putida cannot really grow in the absence of oxygen. So we have two problems here. To convert Pseudomonas putida that is an aerobe into something that is an anaerobe. First of all, we need to produce more ATP. Well, you look at the ways that Pseudomonas putida uses to produce ATP. As I mentioned at the beginning, it doesn't have a real glycolysis. it has an Ender-Dodorov pathway that produces less ATP than the standard glycolysis. So part of the problem can be that we don't produce enough ATP, and perhaps we can generate more via substrate level phosphorylation. Well, we'll see. I'll tell you in a moment. But the other problem, and this is a serious problem, is that by growing cells in the absence of oxygen, then you accumulate a lot of NADH, and therefore you badly need to regenerate NAD. Well, how can you do that? Is something that we try to figure out as follows. By looking at the genes, and this is something that you really have to sit in front of the metabolic map and concentrate and contemplate a little bit what is there and what is not there, we found that Piputida has an phosphotransacetylase able to convert acetyl-CoA into acetyl phosphate. And that's it, period. And the enzyme is there because when you knock it out with a transposon or something, then the activity disappears. You can measure that biochemically. So we thought the following. We can convert acetyl-CoA into acetyl phosphate. What if we knock in the cells the activity that is missing to bring about formation of ATP through a substrate level phosphorylation? Okay, so what we thought is that, well, if we take acetate kinase from E. coli and put it in a vector and put it in Pseudomonas putida, then we can generate acetate and at the same time convert ADP into ATP. So by doing that, very, very simple operation, we can increase the endogenous level of ATP. So we made some constructs with our vectors and so on, and then bingo, we were able to see that by re-engineering or by knocking in the one enzyme that was missing into this pathway for phosphorylation of ATP, concomitant to the production of acetate, we could really increase the concentration of ATP. Well, measuring production of acetate was easy, and you see a big difference. The wild-type strain does not produce acetate, or very, very little that you can detect. And when you knock in this acetate kinase, then indeed you get a lot of acetate, you get acetate. But then when you measure ATP, you observe that, well, you get, with the full-fledged system, something like 20–25% more ATP than the wild type. You may say, well, 25% is nothing, it's just a very little thing. However, let me ask you that metabolism is like economy, and you cannot measure a metabolism by the same token or by the same criteria that you measure, for instance, gene expression levels. So 25% is not a lot, but if I tell you that your salary will be increased by 25%, you will be very happy. So that means that a 25% in a metabolic system, in an economic system, is a considerable difference. But that was not the end of the story, because a part of increasing a little the concentration of ATP, we figure out a way to regenerate NAD. And to do that, again, we thought, well, the cells produce pyruvate as part of the metabolism. What if we enter some way of converting pyruvate into acetaldehyde and then convert it into ethanol in such a way that at the end you regenerate NADH into NAD? And to do that, you need two enzymes, one of them called pyruvate decarboxylase and the other called alcohol dehydrogenase. That you see that the final products are ethanol and NAD. So, well, to do that, you go to the best available source of these two enzymes, and we found, by looking at the literature, we found that this microbe called Pseudomonas mobilis is an obligate anaerobe used by Mexicans for production of some drinks, and it has two very, very powerful enzymes able to convert pyruvate into acetaldehyde and then this into ethanol and bring about exactly the reaction that we were after. So we said, well, we'll try our chances, and what we do is that we construct an artificial operon in which we express under the control of an inducible promoter these two enzymes that are required for ethanol production and also for regeneration of NAD. And then, well, we measure some biochemical, say, parameters and some biochemical compounds. So we could observe that the wild-type strain produces no ethanol, of course, and has none of these two activities, but you have this synthetic operon, and then you induce it, then even if you don't induce it, you get already a signal that things are working well. And if you induce it further with IPTG, then you observe that all the enzymes are there and also that ethanol is being produced. Okay, so in principle, we have something that is better than we had before. At the beginning, we had a bacterium that was completely aerobic. Now we have engineered a couple of tricks that in principle are expected to make this bacterium to be happy under anaerobic conditions. So what we made was to make another synthetic operant with the 3 genes expressing all the activities that were necessary for increase in the concentration of ATP and also for regeneration of NAD. We made sure that all the enzymes are there. We measure them biochemically. And then, well, here comes the critical experiment. What happens when we express these 3 enzymes in P. butida? Are cells now happier under anaerobic conditions? Well, the answer is yes, and you may see in the graphic to the left that cells are quite viable when they are expressing the genes that I just mentioned. When you don't have any of these genes, then the viability grows comes down very quickly. And then depending on the enzymes that you enter, then cells recover more or less viability. But then the ones that really makes a difference is the one where we express the 3 enzymes that are necessary for production of ATP and regeneration of NAD. And also if you look at viability with propidium iodide, for instance, then again, you notice that cells that are not treated or are not expressing these recombinant genes are quite in bad shape after a while. They can incorporate out of propidium iodide, and the cells that are endowed with these 3 activities then are doing very well, and they are not stained by propidium iodide. So that means that we are doing well. Now, another sign of vitality is to put in the cells that we have engineered to be anaerobic a reporter that will indicate that cells are basically happy metabolically. And this reporter is a fluorescent protein that depends for its activity on FMN. So what we checked is that when cells are grown without any extra genes that allow to grow them or to be happy under anaerobic conditions, then the activity of this protein is very low. But when the cells express the genes that we predict that will work, then you see that whether they have oxygen or not, this is an indicator that cells have a considerable metabolic vitality. So we are doing well. So we have now a bacterium that is able to run an efficient metabolism under anaerobic conditions. But we wanted to have that for a purpose, and the purpose was to degrade something that is an environmental pollutant. And that's something, I mean, you can think on various compounds. The one that we use to recheck the concept was this compound here that is the chloropropene that has this structure. That is an actual soil pollutant because it's being used by the chemical industry for various purposes. So this compound is recalcitrant. There are very few bacteria that can degrade it aerobically, and there are no bacteria that can degrade it anaerobically. So the challenge here was to engineer a bacterium that could degrade this compound under anaerobic conditions. So we'll see how it goes. So again, we look at the literature, and we found that there's one strain called Pseudomonas pavonaceae that had been described to run a reaction, a complete metabolic pathway for degradation of this compound. But this strain is completely aerobic, so that means that this reaction cannot be run under anaerobic conditions. So what we made was to ask the genes, or to ask the strain to Dick Jansen in the Netherlands, and then on the basis of the sequences, we constructed yet another artificial operant that we engineer in one of our vectors in front of a promoter and so on, and then we made various combinations, and then we selected the one combination that produced more chloride. So this is it is important at the end to check the efficiency of a particular construct to make sure that at the end you have a kind of reporter of the efficiency of what you have. So this is very simple. It was fortunate because it's simple to detect chloride in the medium. And then, we just went to the ones that had more activity. And we could really see that the chlorination activity was dependent on addition of IPTG. So everything more or less fit, and we have here a 3 cistern synthetic operon that runs the whole show here. Now what we made was to take these 3 genes and put them in the anaerobic strain that, remember, has genes from E. coli and has genes from Pseudomonas. And at the end, what we obtained was this strain that has genes from 5 different origins, okay, that were combined in such a way that, on the one hand, makes the cells to be metabolically active in the absence of oxygen, at the same time makes the cells to degrade this compound. And well, what we have is, bingo, the solution. So when you take this strain with all these genes, all the promoters, all the vectors, all the standards, and all the rest of it, and then you see the viability of the cells, then the viability is more or less fine. The chloride concentration goes up, indicating that the product is being degraded. The concentration of this compound comes down, and therefore that means that the system is working very well. And also we see that the viability is basically all right. So the halogenation is coming this way, and the viability is basically maintained throughout the process. So in principle, this looks very good. And to me, this is really amazing, because by making all these type of tricks, we have really created something that nature has not invented before, namely the ability to degrade completely this compound in the absence of oxygen. When you say, well, this is a very little victory. Yes, it's a very little victory, but it's something that nature has not invented before, but we have invented that for nature, and that means that we have really, I would not say, but maybe we have done in that respect better than nature. Okay, you may say, well, this is just wonderful, so let's go along the way. However, ladies and gentlemen, this is only part of the story. Because on the one hand, we have created a new property, but on the other hand, there are things that, applying these standards and applying this standardization and this chassis where we have taken genes in laboratory still don't work very well. So what we have made is to take the genes and put them in a more or less standardized way with promoters, with intergenic regions. In some cases, we put it in plasmids, in others in the chromosomes and so on. And at the end, we have something that does something new. But what is that is not perfect. Because what I would like is not that the compound is degraded. I would like the strains to grow on this compound. And this is something that we have been able to do to this point. Cells run the reaction and do it very well, but they are still unable to couple this metabolic network with growth. So that means that there's something wrong with this, well, there's something wrong, there's something that really does not fit. So to what extent we can start with a biological system, apply these engineering concepts, and just pretending that by sticking genes here and there, at the end we will get something that is completely predictable. Okay, so again, we are in a building of engineering, and the problem of how to make predictable objects that need to know of parameters that we cannot really calculate with precision is something that is not alien to the history of engineering and to the history of technology. So let me argue, and this is a kind of little diversion in my talk here, that at the end the problem is how to make complex structures, complex objects, without really knowing all the details, all the parameters that are involved in the construction. So I'm going to tell you something that we got some inspiration from, and it is the way a Spanish architect called Gaudí constructed this amazing building in Barcelona that is the cathedral of the so-called Sagrada Familia. So I don't know if you have been to Barcelona. If you go to this place, you find something amazing. This gentleman lived by the end of the 19th century. He constructed this incredible building with no computation, no modeling, very, very little mathematics. And you get in, you find that the entire building does not have straight lines. So it's really arches and very, very few straight lines, but you see a very, very intricate pattern of arches and sub-arches that make the building to have a height without, without a given pattern here. So again, you go, this is a very, very strange structure, and well, you can get further details, and this is just amazing. Okay, so the question is, how could this gentleman construct such a building without knowing all the parameters, because it's an integrated structure. So what you do in this part of the building has consequences or what you have in other part of the building. And this is something that is exactly what happens in biological systems. It's very difficult to change a part of the building, a part of the construct, a part of the cell, of the genome, without having consequences in the other side. How can you really fit that? Okay, so what he figured out, and this is one of the great intuitions, is to make what he called string weight models. So what he made was to take threads and weights and make a model in which he was able to put different weights in strategic parts of the strings and then put everything down in such a way that the very weight of the system would give a solution to the problem of integrating complex structures and complex elements in a building of this type. So what he made then, and this is one of the reproductions that you can see in the museum in Barcelona, is when you have this structure, what he made was just to turn it down. And by turning it down, then you can now see that the arches, reconstruct the arches, the angles of the columns, and all types of details that you cannot really calculate with the formalisms that were available at that time. So we have here one example that to me is very inspirational on how nature can provide parameters that are necessary for the construction of complex systems without having to calculate them from first principles, by letting the system to find its own solution by what you may call natural means. Obviously, this is 19th century architecture, and now architecture is very scientific, and perhaps this is not necessary, but there's a period of time, and I would argue that this is exactly the type of the period of time that we are at the moment in our history, in which we can construct part of a complex biological system on the basis of first principles, but there are other parameters that we don't have yet the knowledge or the formalisms that are necessary for their calculation, and therefore we have to rely on other tricks and then ask nature to provide these parameters, because for the time being we cannot calculate them. So this is the kind of proposition that we are trying to explore in our laboratory, how to calculate optimal parameters for the construction of a complex biological device without being able to calculate them from very, very basic references. So we argue that, well, you can start with some basic elements, but then you have to let the system to evolve towards optimality. This could be one solution. The other solution, and this is, well, obviously for getting these parameters, at the end you need to have some type of evolutionary system in your laboratory in which you just fix an objective in your evolving system, and then you select the combinations that will fulfill that objective. So this is a combination of evolution engineering that is not alien to the history of engineering and to the history of architecture. So it's fine that we bring it to the realm of genetic engineering in microbes. But then we have another trick, and this is something that is turning to be very, very exciting, and it's the following. Sometimes it is difficult to assemble a pathway with all the right parameters in a single cell because of a number of problems. Sometimes the background of the cell, the metabolic background, is not sufficient, or is, some of the intermediates are toxic, or the cells are not sufficiently adapted to host this particular construct. So we wondered time ago whether, instead of having a pathway being recreated on the basis of steps like that, we could have something similar in which each step of the pathway could be brought about by participation of a different cell. So instead of having a single performer, what we may have is a collection of partners, and each of them might contribute to a given process as individual cells. So in such a way that now this very same pathway is implemented through the participation of different cells. So we have two problems here. We have various problems. One of them indeed is how to bring together cells that maybe in nature are not used to be together. So the practical question is whether we can engineer bacteria to stick to each other and to form consortia with a defined stoichiometry, and if possible also with a defined architecture. And to do that, what you need, or we need, is to have adhesins, things that you put on the surface and force bacteria or microbes to stick to each other with a given pattern. So, well, this is something that, again, has kept us busy and entertained for some time. And then we have now a collection of systems for displaying on the surface of Pseudomonas putida various proteins with different activities, including adhesing activity. And one of the anchors that we have used to this end has been a type of proteins that are called autotransporters. And one of the most typical autotransporters comes from Neisseria, and the Neisseria IgA protease that has been known for some time, that is secreted into the outer membrane and gets inserted into the outer membrane through an intermediate situation where an autotransporter domain is bound to the so-called passenger domain that gets exposed on the surface. So the idea that we figure is that if we can use this autotransporter domain as an anchor to display on the surface an adhesin of some type, and then in other cells we can use something that is recognized specifically by that adhesin, we can force physically the adhesion and the attachment of cells that maybe normally are not used to be together. So these autotransporters, in fact, have turned out to be very useful because this passenger domain can be changed with ease. And in many cases, not in all cases, it can be really fused to this autotransporter domain, a beta domain, and then the whole thing behaves in such a way that this passenger domain is displayed on the surface. So just to start with a very, very easy adhesin or artificial adhesin, what we made was to take a leucine zipper. So leucine zipper, as you know, are these protein domains that tend to dimerize. So by doing that, what we have is a situation where we have a autotransporter domain fused to a leucine zipper, and then this expressed in P. putida, for instance, or in E. coli. So in this case, what we have now is the cell that is coated with a partner of a B-party interaction in such a way that now, if in the same culture we have something that corresponds to other leucine zipper, we bring about this, let's say, intimate contact. Well, this is indeed what happened. What we have here is a Pseudomonas putida that has been engineered to express on the surface a leucine zipper anchor to one of these autotransporters. And what we observe is that when you don't express these autotransporters or you induce them, then cells swim, and therefore they move around or they float there. And then the moment that you induce production on the surface of these leucine zippers, then cells collapse and form a cluster that makes them to precipitate, something that you can measure here by various procedures. So, but this is a simple adhesin, but still very useful. And we have been able to make various partners of leucine zippers where partner A will not interact with itself, but will interact with partner B, and we have their combinations. And at the end, we can really get attachment, physical attachment of different types of cells with a given stoichiometry, not absolute stoichiometry, and also with a given architecture, as I will show you later in a moment. But let's face it, leucine zippers, well, it's not a very good adhesin. So what could be the best adhesins? Because at the end, we want to have a collection of adhesins and matching partners for these adhesins. So we can make these combinations in stoichiometry and also in architecture that I told you about. So the ultimate adhesins, in our opinion, are antibodies. So antibodies have this wonderful ability to be very specific to target antigens. So what if we imagine a way of displaying antibodies on the surface of a bacterium and then displaying the target for these antibodies in a different bacterium? So that means that at the end, one will recognize the other and the two will get attached as we wanted at the beginning. But obviously we cannot take the entire immunoglobulin, it's too big, but fortunately there are various procedures by which you can reproduce the business part of the typical antibody as a single chain antibody that is simpler, and all you have to do is to assemble them through various genetic tricks that I will get into the detail, and you have a smaller say piece here that maintains the antigen binding properties of the whole antibody. So what we made, and this is work that we made quite a while ago, is to make fusions between single chain antibodies and these autotransporters that I mentioned before, and display them on the surface of gram-negative bacteria. And what we observed, and it was the good news, is that the protein was indeed expressed and put on the surface, but the bad news is that cells were very unhappy. When cells are unhappy, they start proteolizing the implant that you want them to express, and then things don't work very well here. So, well, what can we do? So what we did was to resort to a different type of antibodies that are produced by camels. So this is the standard antibody with the heavy chain and the light chain. In the camel antibody, what we have is only a heavy chain, and the business part of the antibody is composed of only one single domain called the VHH domain that is very simple and can be expressed in bacteria with no problem. So you need camels, and well, we had to find a camel farm. So we went to the Canary Islands, and once in a while we make friends with the camels, and then we punch them and we get blood and we get antibodies, and we have now a whole platform that allows us to go to a camel, punch antigens, recover the blood, and then we have developed a large a collection of vectors that will allow us to take the sequence directly from the RNA of lymphocytes and produce them displayed on the surface of bacteria. And this is something that in our hands has been very useful. But the important part here is that unlike the case where you have a single chain antibodies with two domains and so on, that is very complicated, in this case cells are a lot happier. So this is what you have, where you have a single chain antibody. Cells, you can see this proteolysis, cells are not very happy here. When you have a single chain antibody, look, cells produce the proteins as a single band, indicating that they are quite fine. If you make a trick and you delete the disulfide bridge, then single chain antibodies can also be secreted. But for most purposes, what is useful is to rely only on camel antibodies. So what we discovered time ago, someone in my laboratory, is that in fact you can display not only one single chain antibody from camel, but also even to make a rosary of various antibodies, antibody parts from these camels, and then they all are expressed, they all are secreted, and they are all active. So this is really fantastic, because what happens now is that you can take the surface that you have smoothed out with these tricks that I told you before, and then we can use various anchors on the outer membrane where we can put, well, either single chain antibodies, but as I said before, this is not very efficient, but we can have a camel antibody or two camel antibodies or more antibodies, and each of them with different specificities to recognize a different antigen. So this is something that is really fantastic, because this is now allow us to make not only combinations of bacteria with different, say, stoichiometries, but also to make what we call origami. So you can take bacteria, you put these adhesins in strategic places, either in the poles or in the center, because we have ways to deliver these antibodies fused to membrane proteins that will go to the tips or to the center of the cell. And then by doing that, we can construct consortia that have an architecture that we think we can predict, we can engineer from very basic principles. So our mission is to end up with something like this, that we call catalytic origami, where bacteria are used as components of an object, and if we endow each of the bacteria with a different activity, we can really amplify tremendously the possibilities that we have to engineer useful objects for biotechnology. Again, this is something that nature does not do. We can, in this case, be doing better than nature. Okay, the other problem that we have is not only to have the two, or the three or the four bacteria together, we have to make them to communicate. So this is one more turn of the screw, how to not only to force bacteria to be together, but how can we force them to communicate. So I will spend the last few minutes of my talk just telling you about a work that is going on in my laboratory, trying to develop a communication language between bacteria in a consortium based on metabolic signals. And well, first of all, let me tell you that bacteria don't speak, obviously, but they communicate through chemical signals. So chemical signals are emitted and they are interpreted by transcriptional factors that use these chemical signals as effectors that result in inhibition or activation of transcription. So this is something that has been observed, is known in literature for a while, but what is really amazing is that one can describe this simple operation of a chemical being in contact with a transitional factor can be described faithfully in the shape of a Boolean gate, a logic gate. So logic gates, again, this is an engineering building, so I'm happy talking about logic gates in this context, are formalism through which the gate will have an input and an output, and depending of a logical operation that is imposed on the system, then you have a deterministic decision. So you have various types of logic gates. For instance, you have an amplifier with the input and the output have the same side, or you have an inverter, or you have an AND gate, and many other gates. And what we have observed, and this was the initial work of Rafael Silva Rocha in my laboratory, is that you can go through the entire transcription literature and translate each of the transcriptional regulation nodes into one or more logic gates. And this is really great because logic gates are the basis of computers and language and artificial language. And this is something that we are trying to exploit exactly to engineer this communication between bacteria. So, well, I don't have the time because the chair will kill me if I get into much detail. But what we have made, and this is something that has been very rewarding, is that every complex network that we observe in microbes can be really described faithfully and rigorously as a collection of logic gates that can be used on the one hand to explain some interesting regulatory phenomena, but can be used also as a source of logic gates that we can rewire genetically and give rise to this communication language between various bacteria. In this case, what you have is the what we call the logicome, the whole of logic gates that rule expression of one very, very well-known system for degradation of toluene and silane in soil that is encoded by the so-called the toll plasmid, that is one of the plasmids of Pseudomonas putida. So, what we have made, and this is the work of Belén Calles, is to start with some of these gates and manipulate them in such a way that we can enter more inputs, more outputs, and make the system to behave again in a non-natural way. So one of the initial experiments that we have made to engineer this type of communication between bacteria A and bacteria B has been, for instance, to program one bacterium to produce benzoate when the bacterium is exposed to toluene, and then the benzoate goes into the medium, and then there's another second bacteria, a second bacterium that will have a biosensor called BenR that will recognize this benzoate and will emit light, for instance. Okay, so in this case, we are using a metabolic signal, benzoate. We are not using quorum sensing or other tricks that people have been using in E. coli. We are interested in environmental bacteria. We want bacteria to be programmed to work in the environment. And therefore, we have to rely in simpler signals of communication, such as metabolic signals. So we have observed, and this is great, that you can use, or we can use many pathways that are known for degradation of different types of compounds, environmental pollutants that have intermediates that can be used as signals of communication in complex systems. So this is just one of the examples of communication. So we have various strains here. We have a sender cell, we have a receiver cell, and then the whole trick is that if there is a communication, then the receiver cell will emit light. So again, without going into much detail, the inducer signal that we have is toluene. The toluene is converted into benzoate, and then the benzoate is sensed by the second bacteria and is converted into production of light. So this is very simple, very basic, but we really think that in a short period of time we can have a big, say, alphabet, not only of letters, but also a whole catalog of sentences and small, say, paragraphs by which we can really try to engineer in an artificial way a communication between bacteria A and bacteria B in such a way that by combining this with this gluing, artificial gluing of various bacteria and artificial consortium, we can really again amplify tremendously the properties that we can engineer in bacteria of this type. Well, I'm sorry, I have to stop at some point. So obviously this is not the end of the story. we have a number of projects exploring metabolic engineering, exploring adhesion, exploring metabolic languages, logic gates, and various other endeavors that I hope that will see some result or some benefit in the future, not only for the sake of our own entertainment and for the sake of developing generic technologies, but also for the sake of solving some specific problems that we are quite interested to tackle. So with that, ladies and gentlemen, I just show you the summary of some of our funders there, and that's all I wanted to tell you, but I don't want to finish without thanking you for your attention. Thank you very much.